Hello, my dear students. I know, exams are just around the corner. And this chapter, bioprocessing, it can seem a little tricky, right? But please don't you worry one bit. In the next few minutes, we're going to make this chapter super, super simple. So let's start with a big question, shall we? Okay, so have you ever stopped and wondered, how can one tiny fungus, something like penicillium, produce literally tons of an antibiotic? An antibiotic that has saved lakhs and lakhs of lives. I mean, how is that even possible? Well, the secret? It lies in building a very special kind of factory. You could call it a biofactory for these tiny little powerhouses. So, the first question is, why? Why do we even need to build these factories? Why not just get whatever we need from nature? And, the simple answer is that nature, as amazing as it is, it produces these useful things in really tiny quantities, just not enough for all of us. To get this, you first need to understand that microbes, they make two kinds of products. Think of it like this. Primary metabolites, those are like the microbe's daily doll, roti, and shovel. The basic food it needs to grow and live. They're essential for its own survival. But then you have the secondary metabolites. Now these are the special goodies. They aren't really essential for the microbe's own growth, but man, are they useful for us. Things like antibiotics, they're all secondary metabolites. And this is the main point. This is the key. To treat millions of people with something like penicillin, we just can't rely on the tiny little amounts a single fungus makes in nature. No way. We need to produce it in bulk. I mean, we need to go big. And that, my dear students, is where bioprocessing comes in. So how do we do it? Well, we build them a house. And not just any house. A very special, super high-tech house. Or, you can even call it a high-tech kitchen. This thing is the absolute heart of our biofactory, and it has a special name. It's called a bioreactor. Just look at this. Now, this is a bioreactor. I know, it looks a bit complicated, but its job is actually quite simple. Its whole purpose is to be the perfect, most comfortable, you could say a 5-star hotel for our microbes. It provides the perfect food, the perfect temperature, and most importantly, it's completely sterile. That means no unwanted guests, no germs allowed. It's a VIP-only zone for our microbes. Let's quickly look at the parts, okay? First up is the agitator. Just think of it as the mixer or a blender. You know how your mother stirs the sabzi while cooking to make sure everything mixes evenly? Well, the agitator does exactly that. It's constantly mixing the contents so every single microbe gets its fair share of food and oxygen. Next, we've got the Sparger, a very important part. You know that little bubbler you see in a fish aquarium? The Sparger does a pretty similar job. It bubbles sterile air, which is full of oxygen, of course, into the liquid. Why? So our little microbes can breathe properly. See, they need that oxygen to work hard and make all the good stuff for us. And then you have these things called baffles. Now, if you stir water in a bucket really fast, you see that whirlpool thing form in the middle, a vortex, right? Well, that's actually not very good for mixing everything evenly. Baffles are like little walls inside the tank that break this vortex and make sure the mixing by the agitator is super efficient. Pretty clever, isn't it? Okay, bachcho, time for a quick practice question, straight from your exam style. Let's match these parts to their jobs. Take a second, look at the options, think about it. Ready? Let's check. Agitator, that helps in mixing, of course. Sparger, that provides the air. Baffle, what does it do? It breaks the vortex. And the jacket, well, that helps control the temperature. See, super easy marks right there for you. All right, so now that we know the machine, let's look at the actual process. It's like, you know how a master chef does all the chopping and preparation, the mise en place, before they even start cooking? Our biofactory does the same thing. There's a whole preparation stage, and this is called upstream processing. The entire bioprocess, it basically has two main stages. It's actually very simple to remember. Upstream processing is everything you do before the microbes start making the product. And downstream processing, well, that's everything you do after the product is made to get it out and make it ready for us. So it's just before and after. That's it. Simple as that. So what exactly happens in upstream processing? It's all about preparation. First, media formulation. That's just creating the perfect, tasty food for the microbes. Then, super important, sterilization, killing all the unwanted germs. Next, you prepare a healthy starter culture. It's just like the little bit of starter that your mom uses to make a whole new pot of curd. And finally, you set the optimal conditions. You know, the perfect temperature and pH. All right, so our microbes have eaten their food, they've grown, and they've produced our valuable product inside that bioreactor. The cooking part is done, so now we move to the next big stage, downstream processing. It's finally time to collect our prize. And what happens in downstream processing? Well, it's all about finishing the job. First, you have separation. You have to remove the product from the microbes and all the leftover liquid. Then comes purification, getting rid of all the unwanted stuff until your product is 100% pure. After that, formulation. This just means preparing it in a form we can actually use, like a tablet or an injection. And finally, of course, you have to package it. Now, this flowchart from your textbook, it shows the entire journey just perfectly. Take a good look. You can see the raw materials going into the upstream processing, then into the bioreactor, and finally out through all the steps of downstream processing to become the finished packaged product. It's like a complete microbial assembly line. So, after all of this hard work, what do we actually get? I mean, what are these amazing goodies our microbial factories are making for us? Let's take a quick look. You're going to be amazed. Okay, pay close attention to this slide beta. This one is perfect for your revision, so I'd say take a quick screenshot of it. You can see all the key products here. You've got ethanol from yeast, our famous penicillin from Penicillium, life-saving insulin from a modified version of E. coli, and even lactic acid, which is in your dahi from Lactobacillus. And you know what? The applications are literally all around us in things you use every single day. That clear bottle of fruit juice you drink, a bioprocessed enzyme called pectinase probably made it that clear. The detergent that gets rid of tough oil stains from your clothes, it's got lipase enzymes. And of course, the really big ones, life-saving medicines like statins for cholesterol, cyclosporin A for organ transplant patients, and strep 2 kinase, which is a clot buster for heart attack patients. All of them come from these amazing biofactories. So there you have it. We've seen how these tiny, tiny microbes working in giant factories can solve some of our biggest problems, from making life-saving medicines to creating biofuels. The possibilities, they're just endless. So the real question is, what world problem will you solve using this technology one day? Think about it. Okay, all the very best for your exams, my dear students. You've totally got this.